This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is a Southwestern blot? Answer: a Southwestern blot is a technique used to identify DNA-binding proteins using labeled double-stranded DNA probes. Question: what type of proteins can be identified using a Southwestern blot? Answer: DNA-binding proteins such as c-Jun and c-Fos can be identified using a Southwestern blot. Question: how does the Southwestern blot technique work? Answer: the Southwestern blot technique combines elements of Southern and Western blotting to identify DNA-binding proteins. Question: what is the meaning of the term Southwestern in the Southwestern blot technique? Answer: the term Southwestern in the Southwestern blot technique refers to the combination of Southern and Western blotting, which is used to identify DNA-binding proteins. Question: what are the steps involved in a Southwestern blot? Answer: the steps involved in a Southwestern blot typically include preparing the sample, extracting the DNA and protein from the cells or tissue of interest, running the sample on an electrophoresis gel to separate the DNA and protein by size, transferring the separated DNA and protein onto a membrane, hybridizing the membrane with a labeled DNA probe specific for the protein of interest, detecting and visualizing the bound DNA binding protein using a detection system, such as autoradiography or chemiluminescence. Question: what are the advantages of Southwestern blot over other techniques? Answer: one of the main advantages of Southwestern blot over other techniques is its ability to identify DNA binding proteins specifically, allowing for the detection of specific interactions between DNA and proteins. Additionally, Southwestern blot is a versatile technique that can be used to study both in vivo and in vitro protein DNA interactions. Question: what are the limitations of Southwestern blot? Answer: one of the main limitations of Southwestern blot is that it only detects DNA binding proteins and not non-DNA binding proteins. Additionally, the technique requires a high amount of specific and labeled DNA probe, which can be costly and difficult to obtain. Furthermore, the sensitivity of Southwestern blot is relatively low compared to other techniques such as chip PCR. Question: what is the difference between Southwestern blot and Northern blot? Answer: while both Southwestern and Northern blot are techniques used to study the interactions between DNA and biomolecules, there are some key differences between the two. Northern blot is used to detect and analyze specific RNA molecules in a sample, whereas Southwestern blot is used to detect and analyze specific DNA binding proteins in a sample. Additionally, Northern blot typically uses a labeled RNA probe, while Southwestern blot uses a labeled DNA probe. Question: what is the difference between Southwestern blot and Western blot? Answer: Western blot is a technique used to detect and analyze specific proteins in a sample, while Southwestern blot is used to detect and analyze specific DNA binding proteins in a sample. Western blot typically uses an antibody-based detection system, while Southwestern blot uses a labeled DNA probe. Additionally, Western blot is used to detect the presence and relative levels of a protein in a sample, while Southwestern blot is used to detect the specific interactions between DNA and proteins. Question: can Southwestern blot be used to detect protein-protein interactions? Answer: no. Southwestern blot is specifically used to detect and analyze specific DNA binding proteins in a sample and not protein-protein interactions. Other techniques such as yeast-2 hybrid, co-immunoprecipitation, or pull-down assay can be used to detect protein-protein interactions. Question: is Southwestern blot a qualitative or quantitative technique? Answer: Southwestern blot can be used as both a qualitative and quantitative technique depending on the experiment. When used qualitatively, it is used to confirm the presence of a specific DNA binding protein in a sample. When used quantitatively, it can be used to determine the relative levels of a specific DNA binding protein in a sample. Question: is Southwestern blot a specific technique? Answer: yes, Southwestern blot is a specific technique as it uses labeled double-stranded DNA probes to detect and analyze specific DNA binding proteins in a sample, allowing for the detection of specific interactions between DNA and proteins.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.